Hi everyone, I'm Maria Holland from the University of Notre Dame, and this is an invited talk that was scheduled to be given in the Morphogenesis session of the 2020 March Meeting of the American Physical Society, which was unfortunately canceled this week due to concerns around the coronavirus. So I'm recording this in my office, and I'm going to upload it online so that we can recreate some of the aspects of the in-person conference that was planned. So the title of my talk today is The Role of Physical Forces in Cortical Morphogenesis. And the topics that I want to cover here are, first of all, some introduction about brain anatomy and background so that I can define some terms that I'll be using. And then I'm going to focus on cortical thickness, which is a main topic of interest in my research group right now. So I want to talk about how cortical thickness varies in the brain, why it varies, and then what that variation means. So here are some images of the brain during gestation, shown here from around 22 weeks of gestation up to 40 weeks of gestation or around full term. So what we see here is that in addition to the really significant size changes that are happening as the brain increases in size, we also see that the shape is changing significantly from originally a fairly smooth surface to around full term, and in adult humans, a very highly wrinkled and convoluted surface. So this is another image of the brain. This is actually an MRI of my brain, looking at a sagittal slice of my brain looking from the right. And we can zoom in to look at these two distinct materials that show up on an MRI. So the outer darker layer we call the gray matter or the cortex, and then the lighter tissue interior in the brain is called white matter. And these two types of tissue differ in the types of cells that make them up, their function, their properties, and so that's an important part of our research. As the cortex is folding around the surface of the brain, it forms both valleys, which we call sulci or a singular sulcus, and gyri, which are the peaks or a singular gyrus. And these are terms that I will be using a lot in this talk. So again, the gyri are the peaks and the sulci are the valleys. And then cortical thickness, this measure that we're interested in, is simply the thickness of the cortex, so the shortest distance between the two surfaces, the white-gray interface and the peel surface. And yeah, that's what I'm going to be talking about today. So in our studies of the brain is a hugely complex organ, and so we have to make a lot of simplifying assumptions at this point to be able to model it. So importantly, one of the simplifying assumptions we make is about the geometry. So instead of dealing with this very curved surface, patient-specific, things like that, a lot of the current work in the field is done on a simple bilayered rectangle where we represent the two types of tissue here, the gray matter and the white matter, with different properties. But the geometry is a very simple, often 2D, often flat surface or object. And then our models generally embed the differential growth hypothesis, which is that increased growth in the outer layer of the brain leads to the buildup of compressive stresses, which eventually leads to instability and the wrinkling in the brain. So the first question that I wanted to address in the talk is, how does cortical thickness vary? So it varies throughout the brain. The cortical thickness is not just a number that sort of holds across the brain in between individuals. These are 12 fairly representative adult human brains, from healthy individuals. And here the brains have been divided into around 120 different regions in both hemispheres of the brain, so around 60 regions in the hemisphere that you can see. And each of the regions has been colored by the average thickness of the cortex in that region. And so you can see on this color scale from 2 to 3.5 millimeters, there's quite a bit of variation. There are very thin regions of the brain and there are very thick regions of the brain. But we can zoom in a little bit, or rather remove some of the noise and see some patterns in this thickness variation. So there are patterns of thickness within the brain that tend to be conserved between individuals. And so specifically, what we can see here is that thin regions of the brain tend to be thin in other individuals, and regions that are thick in one individual tend to be thick in another individual. So here I've just selected one very thick region and one very thin region, and we see that they tend to be thick and thin in all of the 12 individuals. More specifically than that, we actually know that it's sulci that tend to be thin and gyri that tend to be thick. So here I have those same 12 brains. On the left, I've colored only the sulcal regions by their regional thickness, and the gyral regions are colored gray. On the right, only the gyral regions are colored by their average thickness, and the sulcal regions are colored gray. So what we can see here, same color scale, is that the sulci tend to be on the thin side and the gyri tend to be on the thicker side. So again, these are the valleys are thin and the peaks are thick. So this is a repeated, consistent phenomenon across brains, and this has actually been known for a very long time. This is not a new finding from our group, but it is very consistent. So this is a histogram showing gyral and sulcal thicknesses in a public database of 564 typically developing individuals. This is the ABIDE Autism Brain Imaging Data Exchange database. And again, each of the regions is a point in this histogram, so there's many thousands of points here. And the difference between these two distributions is very much significant. So this is a consistent trend that sulci tend to be thinner than gyri. So in answer to my first question, how does cortical thickness vary? It's known, again, this is not a new finding from us, but that the cortex tends to be thicker in gyri than in sulci. So my second question, which is more where we're going to get into new things that my group has done, is why does the cortical thickness vary in this way? So why are gyri so consistently thicker than sulci? And this is an old question. In fact, right now I'm working on translating, or having translated, I guess, an article from 1929 by Dr. Sigmund Bach, originally in German, on sort of related to this question. So this work is being done by an undergrad here at Notre Dame, Andrew Fullwider, who is undertaking this challenge of translating an old mechanics and neuroanatomy article. But there's some really interesting information, some really great quotes in this article. So Bach points out that the structure of the human cerebral cortex is considerably different at the bottom of a fold, so that's the sulci, than at the peaks of the turns adjacent to this bottom, the gyri. And these structural differences between the peaks and valleys of folds are found again and again to occur in the same way at every fold. So he's commenting here on the consistency of this pattern of thin sulci and thick gyri. So his hypothesis for why this would be the case is that cells, as they're bending from a flat configuration to a curved configuration, that they prioritize the preservation of cell arrangement and their volume. And so this is a figure that sort of shows his hypothesis. As the cortex is folding, different layers within the cortex will have to change their thicknesses in order to preserve their volume. So there's been some work on this more recently than the last 90 years. And one of the suggestions for why this would be the case is what I call a biological hypothesis. So this sort of suggests that as neurons are migrating outward to form the cortex, which is how the cortex is formed, that some cells might migrate, might be driven or attracted to migrate to one region of the cortex over another. And in that case, if you have more cells migrating to one area, then you would end up with more neurons there, and that would make that area thicker. And then you would have, if you had another area that somehow attracted fewer neurons, in that area would be thinner. And then it's been shown that thicker areas will tend to preferentially end up on gyri. And so that could potentially explain why we have thick gyri. However, in the last couple years, a collaboration I've been a part of has been working to suggest a mechanical hypothesis for this. Basically, that there are differences in the mechanical state of gyri and sulci, and that affects thickness. So in 2018, we published an article in the Physical Review Letters about this hypothesis, basically. So this was work done with my PhD advisor, Ellen Kuhl, collaborator Alon Gorielli, and Sylvie Budai. So what we showed here is that purely mechanical forces of folding are significant to produce these thickness variations. So we did simulations in so numerical simulations in the finite element method, where we had an initially uniform layer that over time, as it grew and wrinkled, naturally bifurcated into thick peaks and thin valleys. We also did stretched polymer experiments, again, where an initially uniformly thick layer bifurcated into thick peaks and thin valleys, both of which you can see here. And there was also an analytical solution, and all of this data put together showed some nice alignment with sorry, lost my train of thought here. Okay, both of these showed some nice alignment with data from human brains. And so what we concluded here was that this uniform thickness, again, in the starting configuration, naturally bifurcates into thick gyral peaks and thin sulcal valleys. But of course, this, even though mechanical forces may be sufficient to ensure thickness differences, they're probably not the only thing affecting cortical thickness. In fact, multiple factors can contribute to cortical folding. So in addition to mechanical forces, also cellular mechanisms and gene expression. So the work that we're doing now that I'm going to talk about for the rest of this presentation is combining the effects of mechanical forces and cellular mechanisms. And so this work is being done principally by my postdoc, Shuolin Wang, who is working to combine aspects of the biological and mechanical hypotheses to create this sort of hybrid biomechanically coupled growth hypothesis. So in this model, our goal is to simulate preferential gyral or sulcal growth. So test the hypothesis that maybe cells are preferentially attracted to either gyri or sulci, and see what that does to the cortical thickness differences. And so we do that by introducing a growth parameter that varies between gyri and sulci in a consistent way. So to do that, we have to identify what we consider to be a gyrus and what we consider to be a sulcus. And for this, we're using the mean curvature, which is shown here as a color map on the surface of this folded simulation. So 0 is this sort of light blue turquoise color. So gyri tend to have these negative mean curvatures, shown in blue on the gyral peaks. And then sulci are shown in the green, yellow, red spectrum as positive mean curvature. So we introduce this mean curvature, which is then normalized, because mean curvature is a dimensioned quantity. So we normalize it by multiplying it by the characteristic length of the simulation, which is the layer thickness. And then use that to distinguish between gyri and sulci. So the governing equations in this model, we are incorporating volume change resulting from mass change. And so to do that, we use the multiplicative decomposition of the deformation gradient. So we have F, the deformation gradient, equals Fe, Fg, where Fe is the elastic part and Fg is the growth part. So we define the growth part to be isotropic, and a function of the growth parameter theta g. And then the elastic part is found as the remaining part of the deformation. And only that elastic part of the deformation contributes to stress. So we're using a standard Neo-Hookean material model here, but again, you'll see all these E's on the terms of deformation, so only the elastic deformation is contributing to the stress. So exactly how we implement this this differential growth in gyri and sulci is, again, we're using the mean curvature because it's a useful parameter that distinguishes between gyri and sulci. So we have this curvature-dependent cortical growth model. So the formula here, cortical growth depends on mean curvature, so the rate of growth, the rate of change in this growth parameter is, so it's G cortex, so this is a rate, like a uniform rate of growth throughout the cortex. And then 1 minus R curvature, hyperbolic tangent of the normalized mean curvature. So what this looks like is if we set R curvature equal to 0, then the growth rate everywhere is just G cortex. And it's, of course, not dependent on the normalized mean curvature. And so that gives us the case of uniform growth. On the other hand, we have we can also model sulcal growth by using a value for R curvature that's less than 0. So that's shown here in the blue curves. What we see is that in gyral regions, again, where the normalized mean curvature is negative, the growth rate is much lower than the G cortex. And in sulcal regions, where the normalized mean curvature is positive, the growth rate will be higher than elsewhere than this G curvature. So that's what we call sulcal growth. And then finally, we have gyral growth. So if we set our curvature to be greater than 1, then we will have increased growth in gyral regions and decreased growth in sulcal regions. And so that's the model that Shuolun has implemented, and then these are some simulations that show for different values of this curvature dependence parameter R curvature. So we have sulcal growth, uniform growth, and gyral growth across the screen. And then we can track exactly how the thickness is changing in sulcal and gyral regions during this simulation. So, so this plot shows, again, those 3 cases and tracks gyral growth in red and sulcal growth in blue throughout time. So, of course, initially we have both the sulci and gyri increasing at the same rate up until the point of bifurcation. And then we see this bifurcation where the gyri continue getting thicker and the sulci get thinner. But the exact extent to which gyri get thicker and sulci get thinner is dependent on our curvature parameter, so the amount of and type of preferential growth that's happening. So when we have preferential gyral growth, the gyri tend to get thicker, even more thick than in the case of uniform growth, and the sulci actually thin more as well. And in the cases of sulcal growth, the sulci they get thinner, but then they begin to rebound, and the gyri don't continue increasing in thickness much. We can also, just with a little bit of division, we can track the thickness ratio instead of the absolute thicknesses in the sulci and gyri. And so here, again, we see that even in the case of uniform growth, we will get this thickness ratio much greater than 1, although it does plateau at some point. It won't increase infinitely as folding increases. In the case of gyral growth, as we would probably expect, the thickness ratio will continue increasing beyond that, and it doesn't, at least at this point, appear to plateau. And the sulcal growth does still result in a thickness ratio greater than 1, which is interesting. We're actually trying to increase the growth of the sulci, but they will never get thicker than the gyri. So here we see, even with the sulcal growth, thickness ratio is greater than 1, but certainly not as high as we see in the cases of uniform growth or sulcal growth or gyral growth. So now the question that we're wrestling with is what. So we can model these different types of growth. So cells that are preferentially sensing their mechanical environment and choosing to migrate towards sulci or gyri. But the question is, which of these is most realistic? Which of these matches what we see in the brain the most? That's what we're working on currently right now. This gray bar shows the range of thickness ratio that we see in different lobes of the brain. So using this database of 564 typically developing individuals and looking at the thickness ratio between sulci and gyri in each of the 4 major lobes of the brain, We see values between around 1.1 and I think 1.24, something like that. That would indicate to us that sulcal growth is the type of growth that would be most likely to explain this phenomenon. On the other hand, if we look at thickness ratios on a smaller scale, we see a lot more variation, which is to be expected because essentially the now darker gray bar is just an averaging of this lighter gray bar, so it's going to reduce the variation there. So in some regions of the brain, some small regions, it seems that the thickness ratio can be as high as almost 2, around 1.9. In which case, then it might almost make sense for the case of uniform growth to reproduce what we see in the human brain. We're also right now doing some analysis on ferret brains, on infants, and the evolution of cortical thickness changes in them. So this is really where the work is right now. We're really excited about our simulations and just trying to figure out what is the best data to compare them to draw conclusions about about sort of the decisions that cells are making as they're migrating in the developing brain. So in answer to my second question about why does cortical thickness vary, the answer is we're convinced that physical forces are an important factor in these cortical thickness variations, but that this is likely affected by interactions with cellular responses. And so then finally, the last question I presented is, what do these thickness differences mean? Is this just a quirk, something that we happen to see that's sort of interesting trivia but is not meaningful in some way? And we don't think that's the case. So first of all, cortical thickness is an important biomarker of neurological health. These are just a handful of papers that show investigations into cortical thickness and symptoms of different neurological disorders. So this is here we have schizophrenia, autism spectrum disorder, epilepsy, and Alzheimer's. And then again, I want to return to this 90-year-old paper by Sigmund Bach because I think that he had some really interesting words that are currently echoing my thoughts, or my thoughts are echoing his, perhaps. So he said that architectural differences between the adjacent tops and bottoms of the folds are not evidence that the cerebral function is different. So it's tempting to see these differences in gyri and sulci and try to draw potentially functional conclusions about how they might vary. But he's saying that that's sort of a false hope because it's not likely that the function is different between these regions if it's only a difference in thickness that we're seeing. And he hints at the possibility that we might be able to reveal architectural areas that are truly differentiated by function by eliminating the structural differences due to curvature. So these regions might be different, but it's not only because of these thickness differences that are caused by curvature. And theoretically, if we could eliminate the structural differences that are due to curvature, we would see the ones that remain due to functional differences. So he sort of foreshadows some of the challenges that my research group is working on right now. And he says that this critical correction will be an enormous undertaking. So this is essentially what we're trying to do right now, trying to distinguish to what extent these cortical thickness differences are due simply to the geometry, the physics of folding in the cortex, and to what extent they are they may either between individuals, within a single brain, or between species reflect something important, fundamental, functional difference between different regions. So that's the work that we're doing right now, and I'd like to just acknowledge the group of people that I get to work with. So the top row is my research group. Again, I mostly talked about the work of Shuolun Wang here. And then mentioned also Andrew Fullwider, who did the translation of the German paper. Very much thanks to them. And then if you're interested in some more information, the published paper that I talked about in Physical Review Letters There's the information for it, and then you can also check out our lab website or contact me by email or by Twitter. So thanks very much for your attention.